Thank you very much indeed for the invitation. It is extreme honor and only mildly terrifying to address this audience and to try and explain some of the science behind the advances that we've made in the treatment of lymphoid malignancy, and also, I hope, to give some clinical context to the kinds of topics that Janet has so usefully introduced. So, first work out how the thing works. If we look at the changes in mortality from malignant lymphomas over the last 30 years, we can see that this is one of the success stories of modern oncology. Hodgkin lymphoma, the incidence of which has been largely stable over a very long period of time, the mortality has progressively reduced by around fourfold in the last 30 years, so that the chances of dying from Hodgkin lymphoma are now extremely small compared to what we saw in the 1970s. Non-Hodgkin lymphoma, which is of course an agglomeration of about 20 or 30 different illnesses, has shown a somewhat different pattern. The incidence rose very sharply during the last 30 years of the last century, and in parallel with that, so did the mortality, until the point of inflection that we see here, round about the turn of the century. And the difference at that point was the introduction of monoclonal antibody treatment, the specific targeting of an abnormality on the cell surface, which has really changed the pattern of death from this type of lymphoma over time. And I think it's also salutary to reflect on the fact that the progress in these illnesses, certainly during the last century, depended almost not at all on biological insights. So in our fixation with targeted medicines and personalized medicine, it's important to remember that much of the progress that we've made previously has been on a largely empirical basis. This is the CT scan of a patient with a non-Hodgkin lymphoma with a very large mass of lymph nodes occupying pretty much the whole of the abdominal cavity. And this treatment was carried out some years ago. And what we see in response to simple cytotoxic chemotherapy is the complete resolution of that mass. And this patient has remained well and is cured and back to leading an entirely normal life following the completion of this treatment. So we had already achieved quite a lot without knowing an enormous amount about what we were dealing with. But what's become very clear is that we are reaching the limits of our competence in that respect, and there is an asymptote. There is a limit to how many patients we can cure of these illnesses without more biological insight. At the same time, we also recognize that these cures come at a considerable cost. And particularly in the case of Hodgkin lymphoma, we're dealing with a young population of patients. The average age of people presenting with Hodgkin lymphoma is in their early 30s. And if one has a high expectation of survival and a low age of onset, the late effects of treatment become an increasingly dominant consideration. And the sort of things that we're anxious to avoid on behalf of our patients are the changes in the bone marrow, the induction of leukemia as a result of exposure to alkylating agents, the induction of solid tumors. This is a young woman with a breast cancer which has been induced on the edge of a radiotherapy field delivered for the treatment of Hodgkin lymphoma. And this is an epidemic of secondary malignancy that we've reaped on the back of very widespread use of radiation treatment during the 1970s and 1980s. The pulmonary fibrosis that one sees as the result of prolonged exposure to drugs such as bleomycin, and the coronary disease which we see, this is an early left main stem lesion on the edge of a radiotherapy field, again for a patient who was treated with mediastinal radiotherapy for Hodgkin lymphoma. And finally, and of course very importantly, the infertility that we can induce with chemotherapy. This is a biopsy of an azoospermic testis in a patient who's recovered from the treatment of Hodgkin lymphoma. So as well as wishing to understand in detail the biology of the disease in order to design more rational and more effective treatments, what we also wish to do is to understand the responsiveness of these illnesses as our treatment proceeds, in order that we can minimize the exposure to these kinds of toxic insults for the patients who are likely to be readily cured, but maximize the intensity of the treatment in order to maximize the cure rates for those with difficult disease. And the first step in this way is the metabolic assessment of response. And really our approach to something like Hodgkin lymphoma has been transformed in the last 10 years or so by our ability not merely to visualize the masses getting smaller on a CT scan or an MRI scan with simple cross-sectional imaging, but to start to look at the metabolism of these masses. And Hodgkin lymphoma is an excellent case in point. Very often a large lymph node mass will regress down, but you're uncertain at the end of treatment whether what you're left with is a mass of fibrotic material or viable tumor which is going to grow back and kill the patient. And so we've been able to use glucose uptake measured by fluorodeoxyglucose scanning, PET scanning, to compare the results. So here are two patients, both presenting with bulky mediastinal Hodgkin lymphoma, and what one can see here from the coloration is the intense metabolism taking place in these lymph nodes within the center of the chest in the mediastinum. This, by the way, is the cardiac muscle uptake. This patient has had an extremely good response. This is after 2 months of chemotherapy treatment, and what we can see here is that although there's an anatomical residual mass within the mediastinum, this is completely metabolically inert. It is no longer metabolizing glucose. This unfortunate patient, on the other hand, has persistence of metabolic uptake within the mediastinum. This is an extremely bad prognostic sign. So this patient, we can safely afford to de-escalate the treatment to minimize the long-term toxicity of what we're doing. This patient, on the other hand, in order to achieve a cure, we're going to have to escalate the therapy. So we're starting to get a handle early on in the course of the illness, early on in our course of treatment, about who needs more treatment and who can safely be cured with less. And the prospective application of this in clinical trials has occupied us during the last decade. So these are two trials that we reported. The first one we carried out in the late 1990s, and this was in 800 patients with Hodgkin lymphoma. And what we were doing here was what we tended to do in the late 1990s, which was a relatively crude comparison of various different cytotoxic regimens with varying degrees of complexity. And what you can see is that the overall results were exactly the same, whether you had lots of chemotherapy drugs or few chemotherapy drugs. These results were not bad. 90% of patients were alive 5 years from the time of diagnosis, and in functional terms, that means they were almost certainly to have been cured. However, we had to use radiotherapy as the consolidation of our treatment in around half of the patients because we had no certainty that we had achieved a cure before we had functional imaging available. What I'm showing on the right here is a more recent trial which we concluded a couple of years ago now with an international collaboration where we recruited 1,200 or so patients. But as a result of using functional imaging to determine who was destined to be already cured and who had residual disease, we were able to reduce our use of radiotherapy to just under 5% of all the patients. So an enormous reduction in the likelihood of long-term toxicity, particularly in the form of second cancers, coronary and valvular heart disease. With a much shorter follow-up, admittedly, because it's a more recent trial, our overall survival figures look, if anything, rather better, and they seem to be holding up with more prolonged follow-up. So I think we have a first inkling here that we can start to understand on quite a personal level how well our treatments are working and to modulate the treatment accordingly, both in order to maximize the cures and minimize the long-term side effects. But if we really want to get into the biology, if we really wish to understand at a very detailed level what we're doing, we need to understand the cells that we're dealing with. This is a malignant lymph node with some fluorescent immunostains carried out to differentiate between the cells of the germinal center, the part of the node which is very responsive, which is reacting to the challenge of a pathogen, and the mantle zone, which is where a lot of the B-cell memory resides. And we can use a variety of immunostains to start to characterize in detail not just the normal B-cells of a lymph node, but of course to track back and look at the ontogeny, the pathways of development of all the different types of lymphoma that we have to deal with. And we can use these cell surface markers to delineate the different types of cell within the lymph node, but also potentially as targets for therapy. And I'd pick out in particular a molecule called CD20 and one called CD40, which I'll come back to later. But CD20 in particular has been an extremely fertile ground for targeting therapies. And this was the first antibody treatment for cancer which achieved a license and which was responsible for that inflection point in the mortality curve that I showed you at the beginning. And in dissecting the mechanisms of action of these antibodies, we've learned many interesting things about the biology of the lymphocyte. So this is the CD20 molecule spanning the cell surface membrane. And this is where you can attach an antibody. There's a particular loop in the molecule as it protrudes from the cell, which is readily recognized by these antibodies. And rituximab was the first of these antibodies that we used to treat and to improve the cure rates in lymphoma. And what we found in our laboratory studies, and my colleague Martin Glennie in Southampton is a huge collaborator in this, is that this particular sort of antibody, when you attach it to the molecule, results in an aggregation of these molecules on the cell surface into the lipid raft. So if you do an immunostain looking at the fluorescent CD20 protein, you see it aggregated into these little rafts in the cell surface. Interestingly, if you use a different antibody which sticks to a very similar epitope but which has somewhat different properties, it doesn't redistribute this molecule at all, and it stays evenly distributed throughout the membrane. And we're starting again to get into the molecular characteristics of the different antibodies and the way in which they behave on the cell. And interestingly, this antibody relies predominantly on host effector mechanisms, the antibody-directed cellular cytotoxicity response to cure the tumor, whereas this type of antibody is much better at inducing programmed cell death in the cells. So we can start to get down to a really detailed level of understanding of what it is these antibodies produce. And by doing so, we understand which sorts of lymphoma are most likely to respond to this and which will respond to this. And this type of antibody has just been shown to be significantly more effective against chronic lymphocytic leukemia compared to this one. So we make progress in these ways by going back to the mechanisms and understanding how these things are working at a detailed level. So in terms of antibody therapy for lymphoma, the demonstration of this to me was extremely graphic when we were doing the early trials at the end of the 1990s. And this was one of our patients, again with a very large mass of lymph node disease within the abdomen, completely obstructing the circulation to her legs and making life utterly miserable and completely refractory to chemotherapy, who we treated on one of the early studies of rituximab with 4 weeks of treatment. And here we are 9 months later. The lymph node mass is almost completely resolved. Her symptoms had completely disappeared, and she remained in long-term remission for several more years before dying of completely unrelated causes. So her lymphoma never came back, having been entirely refractory to chemotherapy. And for me, this was a very graphic demonstration of the power of this type of biology. And certainly when one looks across a population, this is a retrospective series of what happened to patients with aggressive lymphoma in the days before we used the antibody treatment, where we could expect to cure around half of the patients alive about 3 to 5 years on from diagnosis. The introduction of simple antibody targeting of the cell surface has notched that up to around about 75%. So big and important differences. And these patients are not simply alive for a few months longer, they're cured where previously they would have died of their disease. So big differences from antibody targeting. Parenthetically, when we were looking at the cell surface and looking at antibodies that we might target to it, the CD40 molecule was another thing that's found on B cells. And when I was a clinical research fellow funded by the forerunner of CRUK, the Imperial Cancer Research Fund, I started work on trying to grow B cells in vitro to see if we could do sensitivity testing and understand more about the biology. And I started using CD40 antibodies to cross-link them because somebody had shown that this was a way that you could culture normal B cells in vitro. And what we found very interestingly when we did this was that they upregulated all sorts of other molecules on the cell surface in response to this cross-linking, which makes the cells more immunologically visible. And we see this not only on B cells, but on specialized antigen-presenting cells, the cells of the immune system, which show antigens to the T cells. And by demonstrating that these second messages were upregulated, we started to wonder whether this might be a way to make the lymphoma more immunogenic, more immunologically recognizable. And again, my colleague Martin Glennie showed in a very elegant series of experiments with mice bearing lymphoma that this CD40 was a very unusual target. Normally, when one does a tumor challenge model in a mouse, you treat as quickly as possible to achieve the best possible effect with the antibody. What Martin and his colleagues found was that the longer you left the lymphoma to engraft in this model, so these numbers here are days after engraftment of the lymphoma before the antibody treatment is given, the longer you left the lymphoma to engraft, the more effective the treatment was, which is really counterintuitive. And this opened up a whole new sphere of endeavor. And the idea that what you might be targeting here was not the tumor itself, but using it as a kind of vaccine. So the more tumor that was present, the longer it had grown, the more effective the treatment, suggesting that this was an immune response. And in a series of experiments, Martin went on to show that this was indeed the case, that what was being induced here was long-lasting cellular immunity, and that you couldn't re-challenge these mice with tumor, it just wouldn't engraft. So we went on to carry out a clinical trial to use this, which we've not long completed, which interestingly shows, again, in vivo, in people, the upregulation of exactly the same molecules on the surface of cells as we see in vitro. And this whole area of using antibodies to throw the switches on the immune system and to target not the tumor itself, but the immune response to it, is something which is gaining enormous currency. And the whole area of cancer immunotherapy and how we modulate this was nominated as Science Magazine's Breakthrough of the Year last year. And this has really come from some of these seminal observations about how we can use antibodies to target switches on the cell surface. To return, though, to the biology of the lymph node and our understanding at a molecular level of the heterogeneity of lymphoma, I'll give you one final example of where I think the field is going and shows you some of the challenges that we're rising to in terms of our clinical research. So these are two samples of lymphomas which look identical under the light microscope. The top sample here you can see stains very heavily with these two particular markers. The bottom sample hardly stains at all with those, but stains very heavily with this one. And to all intents and purposes, they look the same in terms of how the patient looks. They look the same under the light microscope, but you can see that the patterns of protein expression in them are radically different. And certainly if you look at the level of gene expression, we can see that these two types of lymphoma, one is called activated B-cell type lymphoma, one is called germinal center type B-cell lymphoma. Each of these lines here represents a different gene and the level of expression. And what you can see is that the patterns of genes that are turned on in these two populations of lymphomas are completely different. So each vertical column is a different patient. And we can see that these genes are wildly turned on in this type of lymphoma and these in this one. And the reason that this matters is not simply that this is a piece of molecular stamp collecting, but that you have a radically different outlook with the types of treatment that we currently use. So people with the germinal center type of B-cell lymphoma have a really quite good prognosis with the type of treatment we use. The people with the activated B-cell type, very much less so. So we're starting to get into the kinds of areas that Janet was talking about. We can run a 20,000 gene expression array on biopsy samples and start to pick out which are the good ones, which are the bad ones, and perhaps most importantly, to relate this to the pathways which are abnormal in these cells. So we can start to look at all these different targets down the signaling pathway from the B-cell receptor down to the nucleus, many of them represented in these lines here, and we can start to see, to simplify the story a great deal, that the activated B-cell types seem to have an upregulation of the NF-kappa B pathway, a particular pathway regulating nuclear transcription. So by dissecting out the patterns of gene expression, by relating these to what happens clinically, we can start to work out what are the important targets for therapy. And again, this is clearly a challenge for clinical research, but this is a study that we've been conducting over the last couple of years, which I think demonstrates very nicely the capabilities and the capacity of the UK in this area. So in this particular study, we've asked patients who've been newly diagnosed with an aggressive lymphoma to have their biopsy sent centrally to my colleagues in Leeds, the Hematological Malignancy Diagnosis Service, where we carry out a gene expression profile. We look at 20,000 genes and we allocate them according to this algorithm into which type they are, and then we're stratifying the randomization in real time. So between the point of diagnosis and the second cycle of chemotherapy, we can send the sample off, we can extract the DNA, we can do a gene expression profile, and we can have the answer back to the trial center in order to randomize the patient. And this is a clinical experiment which can't be done anywhere else in the world. I know because I asked. To be able to turn around this material is something which the NHS is uniquely good at, to be able to centrally refer the biopsy material, to get the answers back, to do the bioinformatics, is something of which I think the UK system can be enormously proud. And what it does is it gives us the kind of platform to answer these questions that Janet was posing in her talk. How are we going to bring the detailed molecular understanding of illnesses such as cancer? How are we going to bring it forward? How are we going to investigate it in the clinic and use the kind of targeted treatments that we want to see brought forward, targeting the understanding that we've made? So I'm enormously optimistic about the future, and I think the UK in particular has a huge role to play in this. I will conclude by thanking my collaborators, who are, the naming of whom would take me at least the 15 minutes I was allotted, but many clinical trialists, laboratory colleagues and trials offices, the funders, particularly, of course, Cancer Research UK, and most importantly, the patients who take part in these studies. This is one of my patients with Hodgkin lymphoma who was in the trial of minimizing therapy, who clearly, having found my chemotherapy not dangerous enough for her liking, took herself off to Everest Base Camp to show how much she'd enjoyed that. Thank you very much.